NYU Langone health surgeons have successfully transplanted a pig kidney that was genetically modified. The kidney continued to function efficiently for 32 days in a man declared deceased by neurologic criteria. The man's heart was kept beating through ventilator support. This is a noteworthy milestone as it marks the lengthiest period of time in which a gene-modified pig kidney has operated in a human being. It is a significant step towards the realization of an alternative, sustainable source of organs for transplantation. On July 14, 2023, a procedure was conducted under the leadership of Robert Montgomery, Maryland, D.Phil, who holds the H. Leon Pachter, Maryland Professor of Surgery position, chairs the Department of Surgery, and directs the NYU Langone Transplant Institute. This marked the 5th instance of xenotransplantation at NYU Langone. The study remains ongoing and will persist until mid-September 2023, with further observation to be conducted. According to Dr. Montgomery, this research displays that a pig's kidney, with only a single genetic alteration and without any experimental pharmaceuticals or apparatus, can perform the role of a human kidney for a minimum of 32 days without being refused by the body. The doctor, who completed the world's initial pig kidney transplant with genetic modifications on a recently deceased human on September 25, 2021, followed by a second procedure on November 22nd of the same year, made this statement. Additionally, the Transplant Institute's surgical team executed 2 pig heart transplants that had been engineered genetically during the summer of 2022. The primary obstacle to overcome in xenotransplantation is the phenomenon known as hyperacute rejection. This event typically occurs within minutes of connecting an animal organ with the human circulatory system. At NYU Langone, researchers have successfully circumvented this issue by knocking out the gene responsible for encoding alpha-gal, a biomolecule that triggers rapid antibody-mediated rejection of pig organs by humans. This modification has successfully prevented immediate rejection in all 5 xenotransplants conducted. Additionally, to prevent novel immune responses, the pig's thymus gland, which is responsible for educating the immune system, was embedded beneath the outer layer of the kidney. This combination of modifications has been proven to effectively prevent organ rejection while still preserving kidney function. In order to ensure that the individual's kidney function was solely maintained by the pig kidney, both of the recipient's original kidneys were surgically removed. Following the procedure, one of the pig's kidneys was transplanted, and remarkably, it immediately began producing urine without any signs of hyperacute rejection. During the observation phase, clinical staff in intensive care closely monitored the pig kidney's performance, conducting weekly biopsies for sampling. Throughout the study, levels of creatinine, a waste product in the bloodstream that serves as an indicator of kidney function, remained in the optimal range, and biopsies revealed no signs of rejection. The procedure was the most recent addition to a more extensive investigation that was sanctioned by a designated research ethics committee at NYU Langone. The surgery was carried out with the consultation of the New York State Department of Health. The significance of this research, according to the leaders of the study, cannot be overstated as it has the potential to save countless lives in the future. The generous donation of the body of a 57-year-old male, whose organs or tissues were deemed unsuitable for transplantation after a declaration of brain death, made this research possible. Livoni, the non-profit organization operating in the greater New York City region that facilitates organ and tissue donations, played a vital role in the initiative to provide guidance, encouragement, and teamwork during the process of obtaining authorization from the family of the donor. Recent data from the Federal Organ Procurement and Transplantation Network, OPTN, indicate that the waiting list for a transplant in the United States has over 103,000 individuals, with approximately 88,000 of those waiting for a kidney. In 2022, around 26,000 individuals received a kidney transplant. In contrast, there are close to 808,000 Americans suffering from end-stage renal disease. According to Dr. Montgomery, who underwent a heart transplant in 2018, there is an acute shortage of organs available to those in need. This scarcity of organs has led to many fatalities, and therefore Dr. Montgomery believes that xenotransplantation is a feasible solution to the problem. In order to perform this procedure, the kidney and thymus gland were sourced from a GAL-safe TM pig, an animal that has been genetically altered by Revivicor Inc., a subsidiary of United Therapeutics Corporation. In December of 2020, the US Food and Drug Administration, FDA, gave their seal of approval to the GAL-safe pig as an alternative source for human therapeutics, as well as a viable food option for individuals who suffer from alpha-gal syndrome, a type of meat allergy that is caused by tick bites. In the past, pig organ transplants that were genetically modified have had up to 10 alterations made. Recent research, however, has demonstrated that a pig kidney with just one gene removed can function at its best for at least 32 days without being rejected. According to Dr. Montgomery, recent evidence suggests that in regards to the kidneys, removing the gene responsible for hyperacute rejection, alongside the use of immunosuppressive drugs that have been approved for clinical use, may be sufficient to effectively maintain the transplant's optimal functionality in humans. This approach may even prove to be effective in the long term. To guarantee the safety of the transplant, the NYU Langone team implemented a combination of standard immunosuppressive drugs and a thorough screening of porcine cytomegalovirus, PCMV, in the donor pig. Research indicates that PCMV can adversely impact the performance of organs and may even prompt organ failure. Fortunately, no trace of PCMV was found after a period of 32 days, and the team maintained diligent monitoring of porcine endogenous retrovirus, PERV, and 6 other viruses that were deemed significant. With the family's authorization, approval from the Ethics Committee, and ongoing assistance from United Therapeutics, observation of the recipient of the pig kidney will persist for another month. The supplementary information obtained during the following weeks will be meticulously assessed to obtain a more profound comprehension of this unparalleled medical breakthrough. Dr. Montgomery expressed that the utilization of a pig that has already been approved for safe use by the FDA, in conjunction with what has been discovered in their xenotransplantation research, brings them nearer to the phase of clinical trials. The team recognizes the potential for this advancement to save a significant number of lives, but they prioritize the highest level of safety and caution as they continue forward. To support our channel's growth and ensure broader awareness, kindly hit the like and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.